Okay, so we covered the nonpolar side chains, the nonpolar R group. There are 9 of them. And then we covered the polar neutral bunch, and you learned the names and the 3-letter abbreviation and the category each they're in. Now we're ready to move on to the polar acidic and the polar basic. Those side chains have a group that behaves as an acid, which means it has a carboxylic acid group in it. That's the functional group it has in it. Or it behaves as a base, so it's got an amine group. Remember, we talked that amines have that lone pair on the nitrogen. They can pick up a hydrogen, and that by definition is a base, if you remember that from intro chemistry. So let's take a look at the names of the amino acids in those 2 categories. All right. In the polar acidic category, we have aspartic acid, not to be confused with asparagine, because remember, asparagine was polar neutral. Aspartic acid is ASP. Asparagine was ASN, remember that? And then there's glutamic acid, again, not to be confused with glutamine. Glutamine was polar neutral. Glutamic acid is polar acidic, and that is GLU. So if I write down aspartic acid, what category is it in? Immediately you know, oh, it's polar acidic. But if I write down ASP, make sure you don't confuse ASP with ASN, okay? The polar basic, there are 3 of them. So polar acidic, there are only 2 of them. Polar basic, there are 3 of them. Lysine, LYS. Histidine, HIS. And arginine, ARG. All right, so let me tell you a little bit about a couple of these. Aspartic acid, like I mentioned a little earlier, with phenylalanine together, they actually make up aspartame or NutraSweet, or that little packet of blue artificial sweetener called Equal. So you basically have an aspartic acid and a phenylalanine and a methanol. They're another extra carbon with an O, and you've got this amazingly sweet substance. The guy that discovered it, it was all by chance. He was just mixing amino acids together to have them as nutrition for patients in a hospital, and he just dropped his sample on the lab table, and as all good chemists should never do, he licked it. At least that's what the story is. And he discovered that the 2 amino acids together make it super, very, very, very sweet, which is something you don't really expect. And that's how this thing was discovered. I'm not 100% sure all that story is correct, but at least part of it's got to be correct. All right, so that's aspartic acid. Remember, aspartic with phenylalanine together form NutraSweet. Lysine, a lot of people will buy lysine from a health food store because, like, if they have a lot of cold sores, they think that this would really help. And it's anecdotal evidence, but it might help because lysine is a very important component in collagen, which is the main protein in connective tissue, like we said in the first video. So it is possible that that lysine could really, really help, especially if their diet doesn't have enough lysine, and it's important for collagen synthesis. So that's something. Histidine, like I told you earlier, the histone proteins are the ones that are around your DNA, and so they have a lot of histidine in them, a lot of this basic, polar basic amino acid. And arginine is actually part of the urea cycle. If you've had any, well, maybe you've studied in biology, but maybe not. But if you've ever worked in a clinical setting and they test urea, well, your body can make urea, which is basically one of the things that your body will do to eliminate extra nitrogen if you have extra nitrogen. One of the intermediates of the urea cycle is this amino acid arginine. Okay, just some information about some of the amino acids. Okay, so now we're ready to put some amino acids together to create a peptide bond. And what is a peptide bond? It is the bond that bonds the amino acids together so that you can make a peptide, a polypeptide, a dipeptide. So when you put two amino acids together, you form a dipeptide. When you put 3 amino acids together, it's a tripeptide. 4 amino acids together, a tetrapeptide. 5 amino acids together, it's a pentapeptide. And then when you've got 10 or more amino acids, that's when we start calling it a polypeptide. Remember, we had a polysaccharide. Now we'd have a polypeptide. Still not called an actual protein, but it is called a polypeptide. Let's start off, though, first with a dipeptide. So what I'm going to do is I'm going to take an amino acid from the nonpolar category. The easiest one is glycine. And then I'm going to take an amino acid from the polar neutral category. May as well take my favorite. So that would be tyrosine. All right. So let me show you how they bond together. So they're both amino acids, right? So they both have a carboxylic acid group. They have this alpha carbon, and they both have an amine. Okay. Glycine happens to just have an H here. So that's it. That's all it has. And the reason it's considered nonpolar is that this H cannot hydrogen bond. The only way an H can hydrogen bond is if it's attached to an oxygen, a nitrogen, or a fluorine. And in terms of biochemistry, it's just oxygen and nitrogen. So if an H is attached to a nitrogen or an oxygen, it can hydrogen bond. If an H is attached to a carbon, it can't hydrogen bond. And if you remember from intro chem days, from whenever you took it, a hydrogen bond is just an intermolecular attraction. I'll talk more about it when we start talking about three-dimensional structure of proteins. But let me just show you what a dipeptide would look like. Okay, so now let me draw out tyrosine. Tyrosine's got a carboxylic acid group, just like glycine, and it's got an amine group, just like glycine, okay? And it's got an H here, but then remember, this category down here is different. So again, you do not have to worry about the structure. I'm going to draw it. Don't worry about it. But here is this thing over here. Can you see? It's got an H attached to an O. Red flag, red flag. The minute an H on the side chain in the R group, if you have an H attached to an O or an N, that means it can hydrogen bond. We're not talking about these guys up here. We're just talking about the side chains. So the side chain, another word for side chain is just the R group. So this R group cannot hydrogen bond. This R group can hydrogen bond. That's why this is polar neutral. This one was nonpolar. Okay? Those were the categories they came from. All right, so what do we want to do? We want to create a bond between the two of them. You guys remember when we created disaccharides, a glycosidic bond, or polysaccharides? We removed an HOH to create a glycosidic bond. When we wanted to put the fatty acids and hook them up with glycerol, we removed an HOH and formed an ester bond. So what are we going to do with the dipeptide? We're going to remove an HOH to create the peptide bond. Okay? So can you see over here? I'm going to show you what I want to do. See this OH and see this H? There's my HOH. I'm going to remove a molecule of water, remove this H, remove this OH, get rid of them. So now you can see, remember we said nitrogen likes to have 3 bonds, carbon needs 4 bonds. Here's 1, 2, 3, needs 1 more bond. So these guys bond together. So this is a peptide bond. So when we have a string of amino acids, a whole bunch of them, one after the other, after the other, after the other, how are they all connected? They're connected by peptide bonds. Okay? So the carboxylic acid of one amino acid gets hooked up to the amine group of the next amino acid. And it just keeps going on and on and on like that. Okay? So let's say I had a tripeptide. What would that look like? Well, let's see. Tripeptide. Let's say I have a tripeptide with aspartic, and let's say I have cysteine, and let's say I have alanine. What if I ask you guys, here it is, what if I ask you, what category is this? You would know that aspartic acid is in the polar acidic. And I ask you, what category is this? Cysteine is in the polar neutral. And I say, what category is this? This is alanine. It's in the nonpolar. So it's going to require a little bit of memorization and practice till you put it all together. So what we're going to do in the next video is we're going to draw these out and see the peptide bonds. Just before I draw them out, how many peptide bonds are found in a tripeptide? Tripeptide means you have 3 amino acids. How many peptide bonds? Well, here's a peptide bond, and here's a peptide bond. 2 peptide bonds are in a tripeptide. What if I had a pentapeptide? Pentapeptide means I have 5 amino acids. How many peptide bonds will bond those 5 amino acids? It would be 4, because 1, 2, 3, 4. Okay? So what if I had a decapeptide, 10 amino acids? It would be 9 peptide bonds. All right? Okay, so the next video, I'll draw this out for you guys, and then we'll be able to get started on identifying the levels of protein structure and the stabilizing forces. Okay?